Red leg partridge now?
Yes.
Okay, thank you.
This is mine. Okay, so good afternoon, everyone. My name is Camilla Broggini. I'm from Italy, but at the moment I'm conducting my PhD at the Wildlife Research Unit from the University of Cordoba in Spain. And today I'm going to talk about the genetic introgression in red leg partridge farms and an open platform system for discrimination between Alectoris rufa and Alectoris chukar. So the red leg partridge is one of the most emblematic game species in southern Europe. The population is suffering a decline due to multiple factors such as habitat loss, agricultural intensifications, hunting pressure, and so on. So to cope with the hunting demand, captive breeding and releasing into the wild have increased. But in partridge farms, red leg partridges are often hybridized with other partridge species, mainly with chukar partridges that have a better growth and adaptivity rate and a better adaptation to captivity. So within the European, national and regional legislation, given this situation regarding the genetic contamination of wild partridge populations, this is to locate ourselves. A program of genetic certification has been developed in Andalusia. So, since the release of hybrids has a significant impact on the genetics of wild partridge populations in Andalusia, to carry out repopulations with autochthonous specimens, a certificate of the genetic quality of the breeding farm and an authorization from the competent authorities are required. So, based on the fact that uncontrolled restocking may lead to conservation concerns due to the high risk of genetic contamination, the main objective of this work is to present the level of genetic introgression in the full cycle farms of Andalusia by applying the program of genetic certification of the Andalusian red partridge farms, and at the same time presenting an improvement of the genetic analysis procedure by using an open platform system that can be freely applied to the aforementioned program. So, since 2011, a genetic certification program has been carried out with the aim of differentiate the farms that produce with genetic guarantees from the others, and of course to adapt the situation of each of the farms to the regulations. Blood samples were obtained from a total of around 9,000 animals from 52 farms interested in participating in the Andalusian genetic certification program. After DNA extraction, the genetic analysis was conducted using the snapshot technology. The presence of the chukar partridge genome in the red leg partridge was detected analyzing 22 species-specific SNP type markers, where a minimum of 17 markers was recommended to detect a level of hybridization equal to 6.25 with a power greater than 90%. So, any farm that in Andalusia wants to release partridges to the field has to be affiliated to the genetic certification program and obtain the certification. So, the program can be mainly divided into 3 phases. The first one regards the application for affiliation to the certification program. Next step is related to the initial representative sampling. And last one involves both the genetic analysis and the compliance with genetic requirements. If so, we can have two different kind of results. If within the sampled populations we have a less than 5% of hybrid individuals and less of the 2% of chukar alleles, then a genetic certification is issued. Otherwise, if within the studied population we have more than 5% of chukar individuals and more than the 2% of chukar alleles, then either a total elimination or renewal of hybrid specimens are required. And so on until obtaining the genetic certification or not. So, among the 22 SNP used, the frequencies of the chukar alleles varied, and on the total of individuals analyzed, the markers with the highest frequencies of chukar alleles were ENO, PLP, and D-loop, while those with the lowest frequencies were CDC2L1, RO1I68, and THB-S1. The percentage of hybridization per farm by province ranged between 0 and more than 50%, with an average being around 23.85%. And out of the 52 farms analyzed, 41 showed a percentage of hybridization higher than the 5%, as reported in the figure by the pink blocks. So among the 52 farms, 29 have been certified meeting the previously mentioned criteria, while 23 have not obtained the certification and therefore cannot release specimens to the field. Although it is possible to analyze partridges from farms, new populations should be analyzed since there may be hybrids into the field. And to eliminate problems related to the snapshot technology, we also present an improvement of the genetic analysis the conventional techniques of genotyping. So with this work, we present the technique, the OpenRI genotyping platform, that is an affordable high-throughput technology based on real-time PCR for analysis of samples with SNPs. The accuracy of the technique can be increased, tend to be increased due to the exchange of the tests, the SNPs that do not work correctly, and optimizing the assays. So we proposed and we utilized 26 genes with described polymorphisms from the literature in order to differentiate the red leg partridges from the chukar partridges. From the sequences of the genes, we designed primers for the amplification of the fragments containing the polymorphism of interest. So, by this procedure, we analyzed 380 samples, equivalent to 4 OpenRI plates, and out of a total of nearly 10,000 genotyping reactions performed, only 36 failed, so representing approximately 0.36% of the total. This figure allows a visual interpretation of our results. So, in detail, the red dots represent samples with both normal rufa sequence for the specific allele for the specific gene. Green dots represent samples with one rufa and one chukar allele, while blue dots represent homozygous mutants with both copies of the chukar allele. So the genetic results found in each partridge farm individually analyzed show that Andalusian farm actually present chukar introgression. 23 out of the 26 suggested markers are currently being used because 2 of them do not appear with a fixed frequency in each population, and one of them was found to be uninformative. And for practical use, we considered a threshold for an individual partridge to be non-acceptable as an Alectoris rufa when at least 2 chukar alleles appear in the analysis. So, along with the chukars from farms, we also genotyped 44 wild partridges coming from field areas, assuming the absence of release of captive animals in those sampled areas, so considered to be pure rufa populations. For free-ranging partridges, our results confirmed that the expectation on the absence of hybrid partridges in those areas prevented from the release of captive partridges, while they showed hybridization in the population with common releases from farms, supporting the hypothesis that actually partridges from farms are the source of hybrid individuals found in the field. Considering that it is quite challenging to distinguish a partridge beyond a first cross generation, for example, through phenotypic characteristics such as plumage, our system shows also a good diagnostic power for identification not only of first generation hybrids, but also of 2 consecutive back crosses with the rufa parent. So to conclude, our results indicate that there is a hybridization problem in the Andalusian farms that release specimens into the field and that it must be controlled. So for this reason, the program of genetic certification of the red partridge farms of Andalusia stands as a very useful tool for the conservation of the native red partridges. However, as I said before, since the snapshot technology presented some technical complications, a more efficient method is proposed to keep on with the genetic control of partridges farm populations. And the system developed here allows the analysis of many individuals at the same time within a short time, and can therefore be applied to the genetic control of partridges, not only in hunting areas, but also in farms before restocking, contributing to the conservation of natural genetic features of wild red partridge populations. And thank you.
Thank you. Very interesting and very important topic. And I have the first question. How do you decide these threshold values of 5% and 2% in the farms?
We have a mathematical algorithm that suggested us to use these values. So we started using those values and applying. Through applying it, we saw that it actually worked. And so we keep on using the same, and of course,
why it's not zero? Why don't you want to avoid totally the ingression of chukar in the farm stocks?
The amount? What do you mean?
Yeah, the amount of chukar genes in the
Yes,
captive population.
actually,
it could be zero, isn't it?
Yes, in the map that I showed before, even if all the regions were around a reddish color, it just means that there are farms without introgression, but the average within the province were always higher than zero. But yes, there are farms, not many, but without introgression.
Okay, I think there will be many more questions. Francis is the first.
This is fascinating. Also with grey partridge, we have the same problem. Not that they are hybridized, but they release partridges that are not locally adapted to the area where they are released. But given how many years, decades, the Spanish hunters are releasing red-legged partridge hybrids, decades, and given how many they release every year, we talk about millions of partridges released in Spain every year. Do you think the certification is coming a bit too late? Is there a hope that by the end of your process of certification, I mean, there is no pure red-legged partridge in Spain. You have to go to England to find some poor ones.
Well, we hope better late than never. And yes, of course, but we have to consider that all the bureaucratic process, we started in like 2003, and then we started like to applying a law in 2008 and then 2009. So of course, it's like it would have been better to start earlier, but maybe also earlier they didn't see the problem, or the problem just didn't exist. We hope that even if by starting maybe too late with this program, we will be able to obtain good results that we are actually obtaining. We are in the process of certificate many farms.
And the second question, those farms who don't receive a certificate, I mean, these are big businesses. They make a lot of money.
Yes.
How are you going to shut them down, or what? I mean, they won't be happy.
Yes, of course. Well, we are still in the process, as the graph show. Like, if they don't obtain it, they are just going to repeat stage 3. So they are going to eliminate part of the hybrids and undergo the analysis procedure again. After I don't know how many times they will need, and at the end hopefully obtaining the certificate, but if not, they are just not going to officially release the partridges to the field.
Officially, yeah. All right.
Another question from my side. You were talking about the introgression of chukar partridge, but you were also talking about adaptations of stocks of the farm stocks to farm conditions.
Yeah.
And do you think that even within the rufa genes, there might be an adaptation to farm conditions, and you, by releasing, even if it's poor rufa genes, you might release a kind of domestic bird there,
Yes.
which is really a problem for wild populations.
Yes, I think that it might be a problem. I also think that the time that they spend within the farm is too short to actually maybe adapt. But I think that this occurs with all the species that are captive bred and then released into the wild. So maybe yes, it might be also this is a problem.
Should be additional regulations for restocking regularly from wild stocks, perhaps.
Yes, also yes, of course.
Yeah.
That's also the idea, I think, of conducting analysis on wild populations in order to use them to reintroduce the animals, maybe in areas where they need to be restocked.
And perhaps it's even not easy to identify these differences on the genetic way. Perhaps you have to even to look at phenotypic changes.
Yes.
For example, number of eggs they lay, and so
Yeah,
I know
course.
from grey partridges that these domestic stocks they lay double the size of the clutch size
Wow.
sometimes doubles, and if you release them, you really can do heavy damage to the
Yes, it's
last breeding.
definitely the same with the red-leg partridges.
Okay, other questions? If not, then thank you a lot. And we are coming to the